Hi everyone, how are you today?
Hi, Katerina.
Hi, everybody. Really excited for the session.
Katerina, just to prepare for the talk, what is the Cas9? Is it has some relation to the Cas9? CRISPR.
Yeah. Oh, Serena, I'm so sorry. I wanted to make you moderator and move to the audience. Please come back up.
I got it.
So sorry. Oh my god. How are you, Serena? I'm sorry, Frank.
Is that what happened?
That
Oh my
happened.
god.
To answer your question, Frank, in the keywords, CRISPR-Cas9 is in there. I pulled this up by using the PMID. That was the quick way I pulled it up.
Sorry, our guest speaker just wrote me an email. I have to respond. I'm sorry when I'm so unavailable. Oh, hi. There you are.
Hello.
Hi. Thank you
Hello.
for coming. Welcome to Science Society. This is Dr. Shiri Levy. Meet everyone. Denise, Frank, Serena, and Jamie.
Pleasure to meet
Hi,
you, Doctor.
everyone.
Hello.
Hello.
Pleasure.
Hello, Shri.
Hi, Dr.
Hello.
Levy. We're so excited to have you with us today.
Very
Oh
excited.
my God, it's so
Very.
exciting to be here because I have never done this before. I'm super excited.
It's a fun
Thank
place.
you. We'll wait maybe a couple more minutes if that's okay with you.
Of course. Yeah. I think like these days, 5 minutes or 3 minutes is definitely normal to wait for people to find where they need to go.
Okay, perfect. Thank you. How was your day so far?
My day was busy. I must say that I was a little nervous to get to this moment. So let's see how it's gonna go.
Oh, I'm sorry that we made you feel nervous, but
I'm
it's
excited.
almost all of our guests because the first time here, so they all feel like you, and then in the end, usually people, I think, enjoyed it. At least that's what they say. Maybe they're just being nice.
And
I'm just,
you'll.
oh, go ahead, please.
Oh no, I'm just going to say once you start talking, and once you know everybody's listening with absolute rapt attention, that you'll find the dialogue just flows nice and freely. This place is really good for that.
Cool. Yeah, moving from being a lecturer or a teacher, I always give talks in front of people, and then through COVID, we moved into Zoom, and now doing this, I love this.
This app became really started and became big, I guess, due to COVID mostly. So yeah.
Well, it's very much a choose your own adventure kind of app, and Katerina has just done such an amazing job at shaping this science society. We just have a lot of great speakers coming in, and I've had a chance to preview your paper, and I'm very excited about the work. It's fascinating.
That is so nice. Thank you. The truth is that I need to get comfortable with the app and see how I can find other people talks and listen to it while I work out or something.
Well, this is a great room to start.
Yeah.
In fact, if I may be so bold, it's the best one to start.
Yeah, I'm gonna bug Katerina later and see how I can get a hold of old or other people's talks. I'm
We
excited
have an
to.
entire archive at your disposal, and anyone
So I go
anyone
to
can
the
listen.
archive. That's where I'm supposed to go.
There are a couple of ways that you can get to the archive. You can access it through Clubhouse. If you were to, for example, scroll to the top of your screen and click on the greenhouse called Science Society, if you scroll down a little bit, there's a section called Replays, and you can see all of the different recordings that we've done with all of our speakers. It takes about 5 minutes or so from the end of the recording for that to be uploaded. So
Uh-huh.
if you wanted to share it very quickly, you could do it that way. There's also, we have a YouTube channel and Spotify. Spotify. And all of
Oh
that
my
is
God,
accessible
this
through
is
our,
on Spotify too.
all of this is accessible on our website. If you go to the website, you can see where all of our different sharing mediums are. We have a YouTube channel as well. So we're
Very cool.
well diversified in terms of options to review the recordings and enjoy the previous conversations that we've had.
And I've just followed you there just now, Doctor. And I'm Jamie, and I'm sure Katerina's followed you, and a few other people I'm sure will as well. And that means that there'll be people that if you're in the app and want to like message someone with any kind of questions when you're just here casually, you can completely reach out. It's a very, very friendly environment.
Okay, sounds good.
Okay.
Thank you.
Yeah, I think we can slowly start. A few more people arrive, and then we can, yeah, more people will arrive, but I think it's fine. So welcome everyone to the Science Society, and we have a very amazing guest speaker here today. We'll talk about her wonderful work, and she got actually Washington Research Foundation Award for the development of de novo protein technology. So that's exciting. Congratulations for that.
Thank you.
Yeah, so just to give you a little bit of background information, our our guest speaker is not just a scientist, but also she was a surgeon combat engineer, force surgeon,
Oh my
Israel
God, you really
defense.
did me up.
I mean, I try. When you take the time to come here, I should at least, know something. It's like, so that's impressive. I'm so impressed by that. And she designed and implemented combat-ready training courses and prepared soldiers for intensive exercise and so on. And Serena here, that is part of our moderators, she's also in like defense research. So there we have something in common. Like, she graduated from the university in Haifa, Israel, and she did there also her research assistant. She was a research assistant there too, and then later she was at the Israel Institute of Technology, Technion, and then she did her postdoc in Seattle with her advisor Professor Hanil Ruhola Baker at the Department of Biochemistry and Institute of Stem Cell and Regenerative Medicine, and she then went to the University of Washington, where she is now, and there she is also an acting instructor and a researcher, and she continues working in stem cell and development and regeneration. And yeah, and as I said, she won this amazing award from the Washington Research Foundation. And yeah, so we are very honored having you here, such an amazing person. And yeah, the stage is yours. Usually somebody like Denise, maybe today, ask you about like a more general question in the beginning, if that's okay, to know you a
Of
little bit.
course.
Okay, great. And then the stage is yours for your research.
Okay. Thank you
Thank-
so much for the introduction.
That was a great intro. Yeah. Thanks so much for joining us, Dr. Levy. We like to ask a question here as scientists. What was the pathway to you becoming a professional scientist? What was the first step in your journey? Can you remember how you found your way into where you are right now? Because we have a lot of young scientists, and it helps to understand the path of people who have already walked it so that they may also do it, perhaps.
Yeah, that is an excellent question. And I think well deserved to ask too. I hate to say it, I didn't choose science. Science chose me.
That's a great answer.
I really, when I was a little kid, I wanted to be a physician. But you know, after I finished my service, and between that and taking all the SATs and all those exams, I failed miserably. And but I did love the science, and those tests really didn't do a lot of justice with me, so I stuck with science. And it was the moment in after I finished my master's degree, when I was at the Technion in Haifa and I started my PhD, I went through a terrible experience in science in terms of the project doesn't work, my hypothesis was wrong, I had to close the project. It was such a tragedy. But I also learned how to be resilient, and I came up with new idea, and I kind of pivot from didn't work and came up with new experiments and new research model and new hypotheses of how to understand life sciences better. And that is when I fell in love with biology and science. And I hate to say it, it had to come with a lot of pain, but I cannot sell you a nice Cinderella story. But this is my story. It came from a lot of pain, and now I completely fell in love with science and biology. And you know, my 5 cents for young scientists is to never ever ever back down to a challenge. You know, that we come up with hypotheses, and many times we'll have to prove them wrong. And the way to go is just to ask different questions. And that's how I started the sciences. So if you want, I'll continue and tell you what I've done in my PhD and how I've rolled into my postdoc.
Yes, if
Yeah.
you'd like to tell us, please.
Yeah, in my PhD, I was working on an archaea that was a ribonuclease, and I identified that in my master's degree. Later on in my PhD, I found a similar family of proteins in human mitochondria, and that was super novel, and I thought it's the coolest thing in the world. So I also characterized that protein in human mitochondria. So I really always worked on regulation and gene expression and control of gene expression in the mammalian system. And I tackled through my master's and my PhD, I tackled all that from the RNA level, and I studied proteins that degrade RNA. And when I came to Seattle to complete my postdoc, I was fortunate to work for and understand control of gene expression from completely different angle, and that's the angle of epigenetics and regulating gene expression through methylation marks. And I'm going to tell you about that today. So overall, I studied a lot of control of gene expression from two different angles in my younger life, from the RNA angle, and now from the chromatin nucleosome epigenetic level. So if that's if we're ready to start, I'm happy to start.
Yes, yes. Let's transition, please. Please begin.
All right. Let me just make sure I don't hear my kids screaming.
Oh, it's
1
fine.
second.
All the time, people are used to it.
Okay, so I'm gonna start by saying good night to all the night owls that are here tonight with us. I know you guys are, I'm not sure, but you guys are based in New York. It's already 9:15. It's late afternoon for me, so way to go, all those night owl scientists, and thank you for having me. As Katerina said before, my name is Shiri Levy. I'm an acting instructor at the University of Washington, and I am part of the biochemistry department and the Institute for Stem Cell and Regenerative Medicine. Just saying all this and just going back to my previous life, the fun fact is that when I was in 10th grade, I had a miserable attempt to be a DJ on the radio, and this whole scenario right now brings me back to being a DJ on the radio, and all I want is to have a whole panel of cool voices and tunes to kind of like do the drum rolls and all that. So that's I'm excited.
What's your DJ name? Do you have one? DJ Cast9?
No, I wish. That would be cool. DJ Cast9. We should definitely start that now.
You heard it here first, folks. It's
That should be a
official.
thing. Yes, yes, yes.
I love it. Well, tonight I'm excited to share with you how we use novel designed proteins to unravel epigenetics and control gene expression in precise loci. And I will talk about the paper that I recently published in Cell Reports. And the title for this paper is Cas9 Fusion to Computer Design PRC2 Inhibitor Reveals Functional TATA Box in Distal Promoter Region. Katerina, is the paper available for people?
Yeah. Yeah, it's up.
Okay, great.
Yep.
Awesome. So I will break down my talk to introduction and mechanism, and the mechanism will include figures 1 to 4, and I'll try to, I have the paper open on my computer, so I'll try to refer to the figure as I go along, and then I'll talk about application of the technology in figure 5 and 6. And altogether, if you guys have questions and want to jump and be like, hey, what does novel design protein means? Hey, what epigenetic means? I'm happy to take a pause and just explain the vocab as we go along. So I'd
Yes,
like
please.
to say, sorry, did you say something?
No, go ahead.
Go ahead.
Say and try to relate every time I start my talk about something that happened to us all the time. So every cell in our body have the same DNA. And if you go through and close your eyes for 1 second and think about all your organs and all your tissues and all the cells inside those organs and tissue, every single cell in your body has the same DNA. And our DNA is 2 meters long, about 3 billion base pairs. However, having said all that, what makes our cells functionally different is epigenetic regulation. And just this fact alone is, to me, mind-blowing. We have brain cells and skin cells and kidney cells, and they all of them behave so differently, and they all express different genes, yet they all have the same DNA. So when I wake up in the morning, I ask myself, how come a brain cell behave like a brain cell and a kidney cell behave like a kidney cell? And what is that in epigenetic regulation that allow a brain cell and a kidney cell to act so differently? So, and this is just normal biology works, but defects in epigenetic regulations are pathological. They affect our immunity, they generate cancer, and they make us age faster. Now, who wants to age faster? No one. So that alone is a really good reason to study epigenetics. The technology we worked on for the first time allowed studying epigenetic regulation at a precise loci, and 3 biological revolutions really allowed it. The first biological revolution is the computationally designed proteins. The second biological revolution is the CRISPR-Cas9 system. And the third one is the biological revolution of induced pluripotent stem cells. So what's true for all epigenetic cells is one complex that trail us through evolution, and this is the polycomb family. The polycomb family, or what we call the polycomb repressive complex 2, which is also known as PRC2. So many times in the talk, I might refer to the PRC2, but this is just a jazz of proteins cluttered together to repress a gene. And the way they do this, those proteins, they work in cahoots, but the active protein, which called the methyltransferase, is called EZH2. And this methylase, EZH2, binds to another molecule, another protein named EED. And once this EZH2 protein binds to EED, it can now methylate promoter regions on a gene to silence and repress those genes. So if you, a biologist, or you ever looked at ChIP-seq data, there is a region of the gene, and on top of it, it looked like mountains of marks. Those are methylation marks, and they cover the whole promoter region on a gene. And the methylation marks I'm referring to are repressive. They close down the gene. They close the chromatin in a way that RNA polymerase II will not be able to move in and transcribe the message. So the PRC2 is an epigenetic repressor. And you can think why it's so important. If we look at stem cells, or if we look at brain cells or kidney cells, we would have to have PRC2 that will silence and block all those genes that shouldn't be expressed at those cells and tissue and organs. Does that make sense? Everything that I said so far?
Yes, it's perfect. Sometimes you
Right.
kind of like are you moving maybe sometimes away from the microphone, or is it just me? Maybe it's my connection, like in
It's
between
not just
your
you. There's
yeah.
Uh-oh.
we're losing you here and there for a couple of seconds. If there's any way to maybe adjust your microphone or your position to go
You have
closer
to
to the
basically
router or
be
where there's a
NASA.
better signal.
You have to
I'm
be
sitting
NASA
on the
precise
router.
holding.
Maybe do you have noise canceling headphones?
I do
Yeah,
actually.
Yeah, it happens the same to me. They don't work well with Clubhouse, so
No headset too.
What?
yeah.
So wait, do you want me to switch to the noise canceling headphones?
No, not noise canceling headphone. Oh,
Oh,
you're using them. Okay. Well,
no,
right
I'm
now
not
we
using
can
them.
hear you fine.
Yeah,
Okay.
just now you sound just great. Yeah. But by the way, Dr. Levy was, I mean, so far so great, and I just wanted to quickly to get the take home learnings that you mentioned. Three important advancement. One is the computational. The second is CRISPR-Cas9. Now, what is? Can you repeat what is the third one that I think I
In
missed?
I call it iPSC, induced pluripotent stem cells.
Got it. Thank you.
So said appears to involve in cell fate determination and developmental transition, and the methyltransferase EZH2 bind to EED to methylate histone 3, lysine 27 to block and repress all those developmental genes. And until now, or even now, for us scientists, when we look at we called histone 3, lysine 27, and 3 methyl marks. In short, we say H3K27me3. Usually on a gene, we would see long, wide breadth of these marks on the promoter region of a gene. And until now, to get rid of those marks, all we can do is take a chemical drug and kind of like erase all these marks from a gene. And that was until now. And what we've done is say, okay, we have all these.
It's actually getting worse.
Yeah,
The beginning of your sentence is muffled.
Okay, let me see. Maybe I should just be on my Wi-Fi. Hang on.
So the key is to not move around that much and to find an orientation of the phone that best picks up your voice and then hold it there almost NASA precise. But
I
just know that Wi-Fi dead spots are about a foot. So if you
okay.
step over a foot left to the right, that might help as well.
I am now down the Wi-Fi, so I should be only on my phone, and that should work. Is that working now?
yeah. Right now you're fine. I'll let you know. I'm so sorry.
Oh, that's okay. The truth is that it's a little stormy here in Seattle, so I'm not surprised. But I'm gonna try and pause if I can.
Right now you sound fine. Like
Okay, good. Okay, so what I was saying that we have these broad 3K27 trimethylation, and until now, in order to get rid of them, get rid of the repressive marks, would mean that we'll turn the gene on and upregulate it and activate it. And the only way to do it so far was to use chemical drugs. And what we were trying to do is stay away from drugs and chemical drugs. And the way to do that was to collaborate with the Institute for Protein Design and design a protein that will block the interaction between EED and EZH2, and by that regulate and activate those genes. Notice that I'm not talking about precision at all. And right now, I'm just introducing to you the work that we've done now 5 years ago, and that was published in PNAS, and characterized this novel protein that is really made from scratch using a computer and AI, artificial intelligence, in order to produce a new protein that is better and greater from nature, and that will allow us to block the interaction between EED and EZH2. And we called it the EED binder. Am I clear until now? Is the connection better?
Yeah, like on my side it's better now. Thank you. Yeah.
Okay, so this binder, this protein binder, mimics EZH2 binding site to EED, but unlike endogenous EZH2, it binds to EED with much greater affinity and function as a biological inhibitor. So we designed a protein from scratch. And back in the day, so that was 5 years ago, it took 2 years. Now it takes 6 months to come up with a new design that is completely brand new. And to have this protein to function as a binder and a protein inhibitor. We also made a negative control protein that has 2 amino acid mutation and that will not bind to EED. So in our PNAS 2017 publication, we characterized the novel design PRC2 inhibitor that is also called EED binder. And what we really learned about the role of PRC2 in human embryonic stem cells, and you can learn more about it if you'll just Google, I think, the EED binder, and I can put it on the chat later too. Overall, the take-home message from that work was that human embryonic stem cells that were induced with the EED binder lost their stem cell morphology, but not the negative control. A pull down assay validated the binding of the EED binder to EED, but not the negative control using mass spec. And immunoblot, which is Western analysis, showed the reduction of global H3K27 trimethylation marks, as well as the PRC2 component, JARI2, and EZH2. And the RNA-seq of this induction of this EED binder confirm the higher expression of differentiation genes and lower expression of the stem cell genes. And finally, ChIP-seq analysis of H3K27 showed the global reduction in H3K27 trimethylation with the EED binder compared to the cells that were not transfected with the binder. So we concluded that human embryonic stem cells need PRC2 in order to maintain their stemness. And so to summarize it, we also proved that the novel design protein, the EED binder, reduce H3K27 marks globally. So now, I told you before that in order to erase all the marks, you would use chemical drug, and now you can use design protein, the EED binder that is made from scratch. So global is great, but local even better. And the better and greater things that we could have done now and think of medicine and precision was to fuse this computer design PRC2 inhibitor, which I called EED binder, to catalytically dead Cas9. So as you know, CRISPR-Cas9 is the new gene editing tool. Cas9 that is not catalytically dead will like a scissors and will be able to make cuts and editing using guide RNA. We use dead Cas9 that basically served just like an Uber, I'd like to say to non-scientists. So it's just a vehicle to deliver the cargo, and our cargo is this novel design protein, the EED binder. So we had two goals in mind. The first one, repress specific site on the gene, and by that activate or upregulate only one gene at a time by removing H3K27 marks at precise loci. And the other thing that we wanted to do is to study which H3K27 trimethylation marks are functionally necessary for gene activation. So we wanted to now not upregulate thousands of genes. We wanted to upregulate one gene and selected genes. What if we wanted to upregulate only 1 or 5 or 10 genes? We now will be able to do it with the catalytically dead Cas9. And so that's the one thing we wanted to know. The second thing we wanted to know is, can we find the sweet spot that is responsible for gene repression? And the way to do it was with the tool that we've created. So if you go to figure 1A, you can see the cartoon, and you can see that normally PRC2 repressing specific genes, and when we fuse EB with dead Cas9 and together with guide RNA, we're able to upregulate specific genes. That was the hypothesis, and that was the working model. In stem cells, we usually generate stable line after making recombinant homology of the construct to the AVS1 locus. And you can see that figure B really described the TET on promoter construct, the EB, which is fused with the linker to that Cas9 and the M cherry. And then you can see the induced pluripotent stem cells. And once we turn on this gene, this ED binder, dead Cas9 gene with doxycycline, now the cells are red. This is true for the ED binder and true for the negative control. What you can also see that when we now induce the cells with this ED binder, there are no global downregulation of EZH2 or K27. And while now we see Cas9 expression, the cells are not downregulating EZH2 or H3K27 in a global level, meaning that these cells are able to keep their stemness. So normally, in order to really understand which marks are important for repression, we tile the promoter region of specific gene. And in figure 1E, You can see how we tile the promoter region. There is guide layout from guide 1 through guide 8, and some of them are closer to the transcription start site, TSS. Some of them are closer away. And now when we do what I called a cocktail transfection with a whole bunch of guides, we can see that the far guides 1 through 8, had very minimal effect. So that's the black bars on the transcription of TBX18 gene. TBX18 is a bivalent gene. It means that it has both H3K27 marks and which are repressive marks and activation marks on the gene. And TBX18 is also sinoatrial markers. The sinoatrial node cells are cells that responsible for the pacemaking in the heart, and those are one of the first cells that are being produced in differentiation in the embryo. And as I said before, they determine the pace of the heart. When we look at guides 3, 6, 4, and 5, we see that now we have a better upregulation of TBX18. So we already had a sense of where the good guides would lay. So when I transfected the same cells, those induced pluripotent stem cells, now with individual guides, now we saw much greater effect of TBX18 upregulation. And you can see guide 5 and guide 6, which are approximately 1,000 base pairs away from TSS, had about 50 to 60 fold increase in TBX18 regulation. To prove that those cells didn't upregulate TBX18 because of differentiation, we looked at the stem cell marker OCT4, and you can see that non-significantly it goes down. We also see through immunofluorescence, and this is figure I, that the OCT4 is not damaged and TBX18 is upregulating compared to the no guide. So this was kind of like the first proof of concept. And what we've learned is that, yes, we can upregulate gene expression only by directing one guide to one locus. But why? What's so special about guide 6? Why is it so important? What was so important about guide 6 region? And this is when we dove into the mechanism of what is so important about guide 6 region, that really the moment you open that site up, now the TBX18 can now be upregulated. So in order to study what happened in the epigenetic point of view, I use ChIP qPCR. So for those of you that are not familiar with ChIP qPCR, what we do after we harvest the experiment, we fix the cells and quench them, and we pull down the chromatin with different antibodies. And here in this experiment, we use the Cas9 antibody, EZH2 and H3K27. And what we've learned is, so right now I'm in figure 2, panel B. What we've learned is that the black bars are the EB dead Cas9, and what you can see that at the mRNA level, we have upregulation of dead Cas9 and TBX18. This is true for the negative control for that Cas9, but not true for TBX18, and there is no upregulation there. And so we proved once again that we're able to upregulate TBX18 at the mRNA level, but what we wanted to know is to understand what happened at the epigenetic level, and for that we used ChIP-qPCR. At the ChIP-qPCR level, we learned that both the ED binder and the negative control are recruited to the site of the nucleosome of the chromatin of where we target the guide RNA. However, when we look at EZH2 and H3K27, we see reduction in EBDAT-Cas9 in those sites, but not for the negative control. So that was a great proof of concept of just seeing what happened inside the cells at the chromatin level, and learning that we were able to downregulate EZH2 and H3K27 at that site, and basically that would contribute to the TBX18 upregulation that you see on the left. Panel D and E really showed that able to target different guides. So this is guide 7, which is really far away. You can see 3,000 base pairs away from TSS. And we didn't see any mRNA upregulation for this guide 7, probably because it was too far away from TSS. But we were still able to look at the epigenetic level and see that epigenetic wise, things kind of stay the same. So, and EBDAT-Cas9 was recruited to the guided area, and EZH2 and H3K27 at that site were reduced. So that just enhanced the question why guide 6 was so successful in upregulating TBX18 when, except from guide 5, no guide was able to do the same job. So before I jumped into the wow of this work, this is a mini, I call it a mini epigenetic memory assay. And we kept the cells, we transfected the cells, and then kept the cells growing at least for a 3 cells division. And we repeated this experiment, and at 5 days, we called that epigenetic memory. And what we saw that the transcript is keep being upregulated at day 5. And then when we look at the epigenetic work, EZH2 and K27 trimethylation marks are still reduced, and EBDAT-Cas9 at day 5 is no longer there, showing that there is some epigenetic memory effect, and the cells are inheriting the remodeling of the marks of that specific region. So far, I tried to convince you that we were able to remodel the epigenetic marks at specific sites. But in figure 3, we kind of like starting to look at the neighborhood around that site and ask, okay, that's great that we've learned that that we can remodel specific sites, but what happened when we go towards the TSS? And we looked at different sites of the epigenome, and we could see that for EBDAT-Cas9, only at 3 days there was recruitment to the site, and that was very specific. But when we look at EZH2 and H3K27, so this is figure 3 panel B and C, we can see a reduction of EZH2 and H3K27, both at 3 days and 5 days, going towards transcription start site, meaning that the chromatin is getting much more relaxed and much more euchromatin compared to heterochromatin, which is a close, tight chromatin. So now the more relaxed chromatin allows RNA polymerase to come in and transcribe TBX18 message. When we look at the other PRC2 components, such as EED, we could see that guide 6, for example, staying put, but this EED is sharply decreasing. This is panel D in figure 3, going towards TSS, as well as GERD2, which is another protein from the PRC2 complex. So far we looked at Cas9 in EZH2 and H3K27, but we also asked, what are those activation marks? Can we validate everything that we see from the repression mark with activation marks? And when we looked at H3K27 acetylations and P300, which is the enzyme that put those acetyl marks for activation marks, are recruited to that same site. And this was true also for RNA polymerase 2 when we looked at day 3 and day 5. And then when we looked at the transcript that's being made, we saw that there are 5' UTR transcripts that are beginning to be transcribed from the area of where we de-repressing the specific nucleosome. One of the things that was super exciting to see was that when we looked at methylation marks now, specifically the marks that are very close to the transcription start site, the methyl marks now gain hydroxyl methyl cytosine marks, and those 5-HMC are now, compared to control, are now upregulated when we transfected TBX18 and guide 6. So overall, we could see that TBX18 is now gearing up for transcription, and this is why we see the transcription. The repressive marks are going down, the activation marks are going up, as well as the methylation marks and hydroxymethylation marks that are now are preparing to being removed. So I kept pitching you the question, why guide 6 is so important? What's so important in guide 6 region that made it such a great hub for activating TBX18 gene? And when we looked at transcriptional elements, one second, let me pause, just one second. When we looked at transcriptional elements, we came up with all kind of ideas, and we looked at transcription factor binding sites. We looked at GC reach regions. We looked at Q quadruplexes that are unique in architecture in that site. And none of those ideas were good. The one idea that came up that we thought of exploring more and more was a prediction of a TATA box. So as you know, TATA boxes are really the initiators for transcription. This is where RNA polymerase see a sequence of TATA and start transcribing the message. And right next to this guide 6, mind you, it's 1,000 base pairs away from TSS. Normal TATA boxes are about 30 base pairs of transcription start site. And what we've learned that right next to guide 6, there is a TATA box, which is, I can't remember exactly how many base pairs away, but dozens of base pairs away from guide 6. And we said, could it be that we sent EB-Cas9 to a specific site on the genome, and it now de-repressed guide 6 site, and now those methylation marks are removed, and now we've exposed the TATA box, and now RNA polymerase 2 is able to read this TATA box and transcribe the message. So in order to prove this hypothesis, what we've done is, if you can see those sequencing file here in figure 4b, we said, okay, if we think that TATA box is responsible for all this, let's remove this TATA box. And we did CRISPR-Cas9. We used CRISPR-Cas9 to really make an incision and really remove this whole TATA box out of TBX18 promoter region. And we made 2 clone. We called it TATA box clone 1 and TATA box deleted clone 2. And in panel C, you can see that the line of EB-Cas9 maintained, and we were able to maintain EB-Cas9 expression. Unlike TATA box 1 and TATA box deletion 2, when we upregulated TBX18, only the wild type was able to upregulate TBX18. But the cell lines where TATA box was deleted was unable to upregulate TBX18 message. So to understand what exactly is going on, we ran a cheap qPCR and we started to pull down EB-Cas9 and EZH2, and we saw that both EB-Cas9 and EZH2, both for wild type and the TATA deleted clones, had recruitment of EB-Cas9 and showed reduction of EZH2. But this is, I think, the biggest wow for me of this paper. We were able to now look at TATA binding protein. TATA binding protein is a protein that bind and lead RNA polymerase to come in and transcribe the message. And what we've learned is the wild type was able to recruit this TATA binding protein. So the wild type has a TATA box. The TATA binding protein will bind to TATA. However, the moment we deleted this TATA box, TATA binding protein was not able to find the TATA box and could not be recruited to TBX18 promoter region. And that's the moment when I've learned that this is exactly the reason why guide 6 is so important. So if we look at the model in figure 4F, you can see that normally PRC2 and guide 6 are covering the TATA box region, which is far away from TSS. And the moment we bring in EB-Cas9, EZH2 is lifted, the H3K27 trimethylation marks are lifted. P300 is now adding acetyl marks to the histone. What you can see that is now this TATA box is exposed, and RNA polymerase together with TATA binding protein and other TATA factors are able to come in and transcribe TBX18 message. And to me, that was so great, understanding the mechanism of how we can utilize EB-Cas9 in understanding some of the elements that we didn't even know that were exist, and now we have a tool and technology to explore all that. So that was super cool, and that's one angle of this story. Katerina, I see that it's already 7 p.m. Do you want me to continue or do you want me to pause?
It's up to you. You can continue. I think we are fine, but
Okay,
you
I'll be
is
super
tired.
breezy now.
Yeah,
Okay, I'll
don't.
be super fast.
Yeah.
So far, I tried to convince you that we understand the mechanism of how this EB-Cas9 works. But since we're looking at one day therapeutics and one day making precision medicine better and greater, we thought how we can utilize this tool and understand if we can make some difference in therapeutics. So now we target a different gene that is called P16. P16 is a cell cycle regulator. It's the I think the name the name of the gene is CDKN2A. It's an inhibitor for cyclin dependent kinase. And what P16 is doing is putting a brake on cell cycle. And in many cancers, P16 is considered as a tumor suppressor. And in many cancer, P16 is highly upregulated with H3K27me3 marks, meaning that it's being repressed and it's being silenced. And what we need to do is really awaken this P16, because since it's responsible to put a brake on cell cycle, and now that cell cycle is being the brake is now being silenced, the cells are keep dividing and dividing and dividing. There is nothing to stop them. So what we've done is, again, tile the promoter region of P16 with different guide RNA. These promoter region was more permissive than TBX18. And what we very quickly learned that not only we can upregulate P16 using EB-Cas9, but also the cell viability of the human embryonic stem cells that we were working on are now being compromised, and the cells are unable to divide because now we've upregulated the brake, and we put a brake on cell cycle control. So again, we proved that we're able mechanistically remodel the epigenetic region of P16 in specific guide. The supplemental also show that we work with diffuse intrinsic pontine glioma. Those are very aggressive cancer cells of kids with glioblastomas. And what we show that we are able to deliver EB-Cas9 to those cells, and with guide RNA are able to upregulate P16 and really block cell cycle. So that's one way how we can use this tool. The other way is to use EB-Cas9 in the stem cell niche and try to now differentiate or transdifferentiate stem cells into different cell type. So anytime in nature, when you go reverse of the differentiation dogma, we call it transdifferentiation. And if you look at figure 6G, you can see that we start from induced human pluripotent stem cells, and we were able to take EB-Cas9 and challenge the question, can we now transdifferentiate induced pluripotent stem cells to trophoblast cells? So trophoblast cells are cells that make the placenta, and create placenta progenitor cells only by epigenetic remodeling. So this time we've made a cocktail of those two transcription factors, and we target guides um, from either TATA3 or CDX2, which are both transcription factors. And this cocktail taught us that specific guides in specific combinations can now upregulate those transcription factors. So both CDX2 and GATA3 were able to be upregulated, while TBX3, which is a marker for placenta progenitor cells, was upregulating too. The RNA-seq that we performed on these cells showed that both guide 1 and guide 5, so guide 1 was better than guide 5, were able to get closer to the placental stage compared to the no guide control. And the coolest thing about this, that we were able to, and this is cartoon L, we were able to take induced pluripotent stem cell, now transfected with transcription factors, and after 3 days, those cells were now primed to become placental progenitor cells. And we continued this differentiation with either forskolin to make synth cytotrophoblasts. So those are multi-nucleated cells, or with TGF beta inhibitor and neurodulin to make extravillous trophoblasts. So those cells are the cells that will latch into the uterine wall and be the connective tissue between placenta and the uterine wall. And we were able to show that cells that were upregulated with that specific guide were able to successfully differentiate into those very specialized progenitor-like placenta cells. And normally you achieve all that with throwing a whole bunch of factors and regulators to take one cell type and make another. But we kind of use a very organic and holistic way to upregulate only specific genes, and in this case, 2 genes that will give us the same result that is, in my opinion, is much cleaner than upregulating thousands and hundreds different genes in the cells that may have off-target effects. So I think that was the paper. I have to give credits to the incredible people that did the work with me, and this is Logesh and Infancia and Diego and Sven, and of course my mentor Hall Baker, that put this work so nicely together. If you go to the abstract and the graphical abstract, you'll now see that you're able to, that we summarize this by really taking the PRC2, creating a novel design binder that is fused to dead Cas9, and now we can specifically tile specific genes and hopefully advance therapeutics one day. So thank you so much for listening. Be more than happy to take questions.
Yeah, thank you so much for this great explanation. It's wonderful. I saw Eli.
Yeah,
Go ahead.
so this will probably be easy for you to dispense with, but I think it's worth asking. One of the things that you found was the distant TATA box. I'm wondering if you looked at its 3D proximity when the DNA is wrapped around nucleosomes.
That is an excellent question. I've never gotten this question before. No, I did not look at the 3D of the TATA box and where it's located in the nucleosome. I'm sure that there is prediction sites that can predict the rotation of the TATA box on the nucleosome. All I know that it was probably in a convenient place for the EB.Cas9 to come in and remodel it the way it did.
So I'm not gonna lie, I'm a little bit confused. It's a lot. So I just want to make sure I'm on the same page. So the
Kirko,
EB is
it's very hard to hear you. Sorry, I don't know if you're on the side.
Are you good? Can you hear me better now?
Marginally. Let's try it again.
Can you hear me better now?
Way better, yes.
Okay. So the EB is the protein that you guys made with the computer, and are you using Cas9 to deliver the EB to the specific site that you want it to get to, or is that
Correct.
okay? Okay, and this EB it was specifically made to bind, or is it specific to a specific place? You know what I'm saying? Like,
Oh, yes. No, the EB was specifically to bind the EED.
oh, okay, I'm tracking.
So the EB stands for EED binder, and it's completely designed to stick and bind to EED.
So, just out of curiosity, what made you guys? What was the determining factor for this is the protein we want it to be stuck to?
Very good question. If you look across many cancer patients, what you see that many of them have overuse of EZH2, and that's the protein that normally binds to EED. And there are many drugs now that targeting EZH2, and that was just a smart decision. What would be the next phase of therapeutics? So the reason why we went after EZH2 EED is because it's such a protein that abundant in cancer.
Awesome, thank you.
Dr. Shaw, you had a question?
Yes, so good evening, Sherry from Seattle. So my question is about in a part of the paper you just mentioned about the gene expression based epigenomic memory and how you just reach out to this goal. And I was just wondering because we know that about the distinct gene expression pattern and it's related with epigenetic cell memory and absolutely transcription factors they can be involved. I was just wondering, did you find any evidence about autoregulation or activity of the chromatin or histone modification or such information?
Really great question. And now that I'm really continuing this research, this is exactly what we're looking at. We're looking at what other proteins are recruited to that site. What other epigenetic modifiers are now changing because of the remodeling that we just force the cell to go to. So these are really good questions, and I don't have answer to them yet beyond what I've shown in the paper. But I'm very curious, just as you said, to learn about the 3D architecture of the chromatin, to learn what other recruiters, writers, erasers, readers are coming into that specific site after we removing H3K27 trimethylation. Really good question. Thank you.
Thank you too.
I had a question. I want to first, highlight or just make sure I get it right. But yeah, no, the work is fascinating in the sense that you've you have a de novo designed protein that allows you to selectively first discover the regions where you can control expression of a particular gene. And it seems that when you find these distal regions, they're much farther than contemporary techniques. That seemed significant. I also have a fascination with trying to interpret the three-dimensional structure, and I was wondering if to turn that question around, when we find a relationship, very distal relationship in sequence from the activation region and the expression of the gene, does that actually tell us something about the structure, or the or how is that could be some other factor going on? But does that actually tell us that these are spatially proximal, even though they're
Yeah,
very distant in sequence space? Yeah, go ahead.
yeah, I think you're absolutely right. And very unfortunately, still today at 2022, and I really hope very soon I'll be wrong. It's really so hard to find techniques that will allow us to understand the 3D architecture of the chromatin. So the available 3D structure that we can use right now are high C. And high Cs are true for mega base pairs long. I'm hoping that this will become very high resolution, where we can look at 2000 base per region and a 500 base per region and understand the loops of the chromatins and the nucleosome orientation in order to understand why genes are being expressed the way they do. That's a really good.
Yeah, could I
And
could I
just
riff on that a little bit, if I could? This is John. I think this is one of the most underappreciated aspects of the genetic code, is that the tertiary and quaternary structures of the DNA folding are critical. So if you look at long repeated sequences that are quote unquote nonsense, we have to understand that some of those will be critical structures to ensure the approximation of the gene itself with all of the various types of influences on that gene, that the approximation of those two in three-dimensional space is subject to mutation. So mutations in dark DNA that affect the expression of a gene in some circumstances, not all, but in some circumstances, are invariably going to be proven to be how they changed the molecular proximity of those two different species on the same strand of DNA, and we're going to find that, particularly unless you do really deep some of the most difficult domains to sequence in DNA is where they're locked in a quaternary folding structure that doesn't lend itself to continuous reads. And so what you just described, I believe, is going to be a gold mine for understanding what has remained fairly mysterious in the dark DNA.
Thank you, John.
Does that make sense to you?
well,
Yeah, and I couldn't agree with you more. I mean, I'm assuming that the gold mine that you're referring to is really the understanding high C and having a high resolution of the chromatin folding.
Exactly.
Yeah, I agree with you. Yes, and I hope that the techniques will become greater and better. And right now, I think it's only CTCF pull down that allow understanding high C. I really hope this will get in much better resolution than what it is right now.
Well, I wonder if there's approaches to attach fluorescent labels, for example, to the safer your your design proteins and regions of the actual expression, and perhaps detect proximity through resonance transfer, those type of techniques. So you could confirm that there's a proximity there spatially. That's one thought. Another thought that I, when I hear about methylation, particularly trimer-methylation, turning a primary amine into a quaternary salt, is it known whether that alters the fold of chromatin, or in some way changes what's buried and what's exposed um, more profoundly than just blocking locally, through either solubility, buried surface area, those type. You've changed the chemical properties quite a bit when you try methylated amine, but that can also have more profound effects on the preferred energetics of its environment, and I wonder if that's driving quaternary changes.
I think that's a brilliant idea, and another way of thinking about it, which would probably yield complementary results, is the application of bispecific antibodies. So bispecific antibodies are used in therapeutics to, on one of the binding sites, bind a drug, and another binding site bind the target. But in this case, you could consider the regulatory species as the drug and the gene as the target. And if you could find where bispecifics were localized when you have engineered them to have a regulatory binding as well as a direct genetic binding, it may be, and then you could fluorescent tag the bispecific at the base. That would be a really interesting probe to look indirectly at the hypothesis that distance in base pairs does not equate to distance in space.
So there are a few techniques that can be used in this regards. I'll just mention CHIP, the chromatin immunoprecipitation, but a couple that are kind of interesting, though, perhaps. Well, one is really interesting, but it's probably not widely established and probably won't be widely established. It's called a DNA nanoscope, and I forget the primary author, but George Church is involved in it, and basically there's they tag things with either recognition proteins or aptamers, and they have a way of measuring how far the two things are by having multiple pieces of DNA hybridize, and then they sequence it, and by how long the resulting sequences are, you get basically a 3D distance map. Now that's a little fanciful. It's neat. It's amazing that it works at all, actually. But there are more modern light microscopy techniques like light sheet microscopy that really can localize labels with precision down to on the order of well under a wavelength. Well under a quarter to wavelength. So I'm not throwing it up because I'm probably going to get it wrong, but it's just amazingly close. And there's actually a whole family of these high resolution light microscopies that have come about in the last 10 years that could probably be brought to bear.
Yeah, the new director at the La Jolla Institute of Immunology, that's her specialty. And they have some amazing technology they use precisely in that way.
Sorry, guys. I'm not sure what happened, but I'm back.
Right on, right in time. I had a couple questions. I
Great.
was wondering, so you use Cas9 in this, where we've dubbed you DJ Cas9. I bought the domain already. We can talk about that later. Just kidding. I was curious why you didn't use Cas12, and also there's great implications in terms of suppressing cancer in your work. So I was curious, do you see that happening in your lifetime, or is this a mission that future generations are going to have to carry out? What do you think?
So first, Cas12 wasn't a thing 5 years ago when I started working on it. Cas12 is definitely up and coming now, and getting more traction right now. So, and yes, the one of the things that we're doing is moving the novel design proteins to other cases and other dead cases. Yes, I see this happening in the next few years. I think that CRISPR will become a therapeutic agent. It already is in some instances, and it's already being delivered to the liver. I think this will become the next phase of therapeutics. Did I answer?
Yes, fantastic. I mean, there's certainly, and there's so many different applications of this type of science to improve human health. So yeah, definitely looking forward to the future in this regard.
Thank you.
Questions? Anyone else have questions? Flash your mics.
Hi, Dr. Levy. I have a quick question on the design of the protein, the EB, the binder protein. Do you know by chance that what software that they is the
Back in
best
the
tool?
day, yes, yeah. So back in the day, this is a collaboration with David Baker's lab. This was used using Rosetta, and now the second or third generation of Rosetta is the Rosetta Fold. And you can Google all that and get all the information about the design.
Thank you.
Yeah, I wanted to comment. We had that I think it's really interesting that you worked on placenta, differentiating the stem cells into placenta cells, because we had the guest speak on Friday that presented his work of ex utero development into E11.5, but not until the end. And they said for this, they would have to create like an artificial placenta, or maybe grow a placenta. So this is perfect. You should collaborate with them on this.
Yeah, I need to look that up.
Yeah, I can share it with you. He's actually in Israel, working there at the Weizmann Institute. I don't know if you know it, but
Yeah, of course.
anyway,
Yes.
that's interesting. And yeah, this is such an amazing work, and I really think that just also the basic science that you figured out to get to this tool basically is amazing. I found especially fascinating the timing and the inheritance part of the of these mechanisms are really interesting. So how long did you ever try how long this inheritance stays, like how many, I guess, passages or how many generations? Did you try if it at some point doesn't get inherited anymore?
So I did try up to 12 days. The problem that I ran into, and this is why I didn't publish it, is that the stem cells are no longer stem cells if you culture them for so long without moving them on a plate. So I think it requires more work The most that I could do keeping the stem cells was 3 days and 3 cell divisions, which worked. The 12 days showed continuous upregulation of the transcript, but I couldn't see epigenetic regulation, downregulation, and I don't even think that we ever harvest those cells, so I can't really speak to it. But the one main problem that we had was in what media we need to condition those cells in order to allow them to become what they want to become. And only later in my work I've discovered that I could buy some kind of and benign media that I can culture the cells. It's all tricky when you work on stem cells. I think that if I would work on different cell type, it might be a little easier to study. The stem cells just drift so easily if they're not kept in their media. So that was some of the challenges studying epigenetic memory.
So I was just wondering, in day 5 or 7, did you, notice any kind of instability, happen?
So at day 5, my control cells were still stem cells, so I could compare the study group to stem cells. On day 7, the cells had already drifted, so it's really hard to have a good reference once the cells are drifting. So I cut the experiment on day 5. But if I had to do it all over again, I would look at a different media that will just be perfect to grow the stem cells and the study group. Or I would just work on cancer cells that always kept on the same media.
It's always interesting because when we get into the technicalities of things, like Katerina was mentioning earlier with the ex utero mice embryology, they were at day 12 was where the limits of their experiment, and you're also speaking about cutoffs and limitations, and so it's always interesting to hear those parallels and wonder, how much further you need to push to get as far as you need to go.
Yeah, science will do that to you.
Yeah, so why I asked the question earlier was I was thinking when you use this, let's say in therapy for anti-cancer, so you could use this to basically suppress further cancer development, I guess. So I was thinking, well, you basically stop them from being cancer cells, so you don't need to care too much about the next generations, right? Am I right? So
Yeah, that's right.
So that's why it doesn't really matter.
For that specific gene, you would just terminate them. But if we were to look at different cell types, one thing that I can think of is, for example, beta cells and islet cells of the pancreas. If we could take beta cells and now retrain them and reprogram them to produce insulin, that would be something that would have an epigenetic memory necessity, unlike the cancer cells that we just want to terminate.
Yeah, exactly. Yeah, that's why. And then so you can be also very precise on the temporal scale, right? You can be precise in theory with the loci, but also when you want them to kick in, or do you need to give the trigger drug basically?
Um, uh, you know, if we're talking therapeutics, is completely different ball game than the work that I've done so far.
Sorry.
I don't think doxycycline is a thing to upregulate, you know, gene expression when it comes to therapeutics. But I'm sure there is all kind of clever ideas of how to express genes in a therapeutic forms.
Well, yeah, maybe we can use something. Maybe we can use something, but it's, I think in general, it's a good idea to have a, so that in theory you can stop. If something starts going wrong, you can just stop
Oh
taking that
yes,
drug and then,
yeah. When you direct it to specific genes, yes. Yeah. I'm sorry, I didn't understand the question. Yes, when we direct EBDAT-Cas9 to specific genes, we can control if the cell, if we activate a kill switch or not.
yeah, I just wanted to point that out for the audience, that this is like another, you know, another very amazing aspect for the future, for future treatment. So it's wonderful. So I wanted to check with you because you've been here over an hour and a half and you talked a lot. How are you feeling? And yeah, this was such a great talk.
I feel so great. Thank you so much for these incredible opportunities. I'm gonna go to the archives and listen to a whole bunch of cool talks.
Actually, a previous guest speaker of ours, she was here because she gave a talk about her neurodevelopmental PhD study, and she went to postdoc position and is going into your field basically in epigenetic and gene regulation. So she came to listen, so that was good.
Very cool.
Yeah, so the Ethan just joined us. Ethan, do you have like a last question you want to ask our guest speaker?
No, thanks though.
Okay, great. Just checking. Okay, does anyone have maybe a last question? Last comment?
Oh yeah,
Yeah,
if I
go
may, I
ahead.
just have one question, the last question, if I may. Yeah. Dr. Levy, I'm curious. As a not in the field, not as a layperson, but very interested in the cool research that presented. Just yeah, really appreciate that. So on the figure 4A that where you show that the the TBX 18 that basically you somehow identify the TATA box and INR. Are these like the Euro suspects, or what led you? I mean, I do search in the text. There's some mentioning of a technique called G quadruplex. Would you be kind to elaborate a little bit? How did you like identify that as the target?
Yeah. So I looked at a whole bunch of transcriptional elements. Some of them were transcription binding sites. The other were the quadruplexes that you're mentioning. And each one of them has its own software and website to kind of like plug in the sequence and see if that's a good hit for either transcription factor binding site or quadruplex chromatin architecture. In order to find the TATA box and the INRs, what we call the initiators, I use the Element software. and of course it's in the paper. You can find it. That is where I plugged in 1,000 base pairs of the promoter region, and I got very strong hits to the TATA box and the initiators.
Great, thank you.
Of course.
Okay, great. Thank you so much again. And come back anytime. And yeah, if you want me to send you links for any talks, or if you want to join us again and listen or be part of the discussion, ask questions, please come. You were such a great speaker. It was just very wonderful having you here. You were a great public speaker.
Likewise. Thank you so much, Katerina.
Thank you. And
Bye.
thank you. Yeah. And thank you everyone for being here, asking questions, being interested. And if you liked it, follow the club, Science Society, and we will have more events, maybe not exactly like this, but we have more guest speakers coming. And tomorrow morning at 11:30 a.m. EST, we'll have Dr. Carl Ernst. He will talk about dimmer switch he found for human brain cell growth. And then we'll have in the evening, then we'll have 2 rooms because of rescheduling, and then somehow it became 2 rooms on one day. Dr. Gallardo, he will be talking about reducing brain damage in Alzheimer's, a recent paper of his. And then on Wednesday, we'll have Unlocking the Code of Sight with Dr. Bruce Hansen about the visual system. And then on Thursday, we'll have in the morning at 10 a.m. EST, quantum physics room. Dr. Kutak he's a nuclear physics professor in Poland. He will talk about the interior of protons and how they are maximally entangled. And then we'll have Dr. Huang talking about his research and showing why most smokers don't get lung cancer. It's a recent Nature publication and will be very interesting. And on Friday, we'll have from Japan a team coming, Dr. Yanigasawa and Dr. Fukuma, talking about how they can read pictures of your mind's eye. So basically what you visualize in your mind. So yeah, it's an exciting week, and we started with this amazing talk. I really loved how you started explaining close your eyes and imagine all your cells to explain epigenetics. That was really, this will stay forever in my mind. That was amazing. So yeah, thank you so much.
All right.
And
Thanks, guys. Take care.
bye.
Thank
Have a
you.
good
Bye.
night.
Thank you.
Bye.
Bye.
Thanks, Sheree. Thanks, Katrina.